Welcome to the Urban Herald. Today we're diving into a genuinely remarkable scientific story. It's this convergence of deep history, cutting-edge chemistry, and just a desperate need in oncology.
It really is. And this isn't about some brand new billion-dollar miracle drug.
No, not at all. It's about a 70-year-old medication. A common blood pressure pill that, as it turns out, scientists just discovered is actually a potent tumor silencer.
It's the ultimate example of something hidden in plain sight. I mean, for decades, this drug, Hydralazine, it was saving lives all over the world, but the exact way it worked was a total mystery. A pharmacological black box.
A black box, exactly.
And now, because of some just exceptional chemical detective work, researchers have not only solved that puzzle, but they've also revealed its potential to treat glioblastoma, or GBM.
Which is one of the most aggressive, most devastating brain cancers we face.
Without a doubt.
You know, I find that so compelling that a key piece of the puzzle was already there, just sitting on the pharmacy shelf. This breakthrough came from a team at the University of Pennsylvania.
A really multidisciplinary team.
Right, and they unveiled the precise molecular mechanism of hydralazine, which you might know by the brand name Apresoline. This research just fundamentally changes how we view this drug.
It absolutely does. It goes from a simple antihypertensive to a potential key in a whole new strategy against this really intractable disease.
So, as part of the team here at the Urban Herald, our mission for you in this deep dive is to unpack this whole story.
We really need to get into the scope of the glioblastoma crisis, look at exactly how hydralazine works at that molecular level, and then talk about why this whole drug repurposing strategy is so critical.
And importantly, we have to talk about the roadblocks, the big ones that are still there.
Oh, yeah. Chief among them, the blood-brain barrier.
This whole story, it's just a validation of that curiosity-driven research. Hydralazine's secret was hidden for generations. It's this powerful, affordable tool that we only just now got the instruction manual for.
To think a drug on the WHO's essential list since the 1950s had this other life just waiting.
It's incredible. Okay, let's unpack this. Let's start with its history.
Sounds good.
Hydralazine's history is pretty extensive. It was actually first synthesized when they were looking for anti-malarial compounds back in the mid-20th century.
Right, and it didn't really work for that initial mission.
No, but it quickly found its true calling, in cardiovascular medicine.
That's right. Since the 1950s, it's been a cornerstone of therapy, which is why it's on the WHO's list of essential medicines. Its primary job is as a potent vasodilator.
Meaning it relaxes and widens blood vessels.
Exactly. It brings blood pressure down efficiently, which makes it vital for severe hypertension, a hypertensive crisis, and especially for preeclampsia.
And preeclampsia is, it's a terrifying condition for pregnant women.
It is. It's this sudden, dangerous spike in blood pressure during pregnancy. It can lead to organ damage, seizures, even death for both the mother and the fetus.
So hydralazine's ability to just rapidly drop that pressure is a frontline intervention.
A life-saving one. This drug has been saving lives for, 70 years now?
But here's the mystery. The thing that's frustrated pharmacologists for all that time, despite its success, the precise molecular target, the exact switch it was flipping inside the cell, that was completely elusive.
It was a true pharmacological ghost. It's a great example of how medicine sometimes just moves forward by application before we fully understand the science.
We knew what it did, but not how.
Exactly. We knew the pharmacodynamics, what the drug did to the body perfectly, but we didn't know the specific molecule it was interacting with. Doctors knew it worked, but the keyhole it fit into was a total guess.
Until now. The UPenn team figured that solving this puzzle might reveal something unexpected.
And it certainly did.
Okay, before we dive into the new science, we really have to just sit with the gravity of glioblastoma. This isn't just an academic exercise. It's about a truly formidable disease.
Glioblastoma is, it's devastating. It's a grade 4 tumor. That's the highest grade of malignancy you can get. It's the most common and aggressive primary brain cancer in adults.
It accounts for almost half of all of them. Right?
Nearly 48%, yeah. And it typically strikes later in life. The median age of diagnosis is around 64.
And the numbers, the prognosis, they're just heartbreaking. We hear aggressive cancer all the time, but for GBM, that word feels, inadequate.
It really is. Even with the absolute best treatment we have, surgery, radiation, chemo, the median survival is only about 10 to 13 months.
So half of all patients diagnosed won't make it a year.
That's the reality. We're talking about a disease where the standard of care is just buying a little bit of time. Less than 10% of patients survive beyond 5 years, and if you leave it untreated, survival is maybe 3 months.
Which is why the stagnation in treatment is just so frustrating for everyone.
It is the defining tragedy of neuro-oncology. The current standard protocol surgery, radiation, and a chemo agent called temozolomide, that was established by the Stoop trial way back in 2005.
2005, so we're talking nearly 2 decades of basically standing still.
The gains since then have been minimal. It just underscores the desperate need for completely new ways of thinking, which is exactly where this kind of drug repurposing comes in.
Let's dig into why it's so resistant. Because the tumor's environment is actually key to this new discovery.
There are a few big reasons. First, cellular heterogeneity.
So not all the cells in the tumor are the same.
Not at all. They have different mutations, different characteristics, so one drug can't kill them all. Second is invasion. GBM cells migrate deep into healthy brain tissue.
Which makes surgery so difficult.
It makes complete removal impossible. You can't chase every single cell without causing catastrophic brain damage. Surgeons get the bulk, but these microscopic tendrils are always left behind.
And then there's the microenvironment itself. The tumor creates its own little ecosystem.
Precisely. And that ecosystem is defined by hypoxia, low oxygen. As the tumor grows so fast, it outgrows its blood supply.
And you'd think that would kill it.
You would, but it doesn't. That low oxygen state is a trigger. It paradoxically makes the tumor even more aggressive, it helps maintain the cancer stem cells, and it makes them resistant to chemo and radiation.
So it thrives in the worst conditions.
It's a hostile ecosystem designed for its own survival. And it's that specific adaptation to hypoxia that this hydralazine research zeroes in on.
So we have this 70-year-old drug mystery and this incredibly aggressive cancer that loves low oxygen. How did Professor Megan Matthews' team connect these two things?
Well, you can't solve a mystery this old with old methods. This required something called chemical proteomics.
Okay, what is that?
It's a very sophisticated field that mixes chemistry, biology, and mass spectrometry. They basically had to go fishing for the one specific protein that hydralazine physically grabs onto inside a living cell.
Fishing among thousands and thousands of other proteins.
Exactly. And their secret weapon, their lure, if you will, was this clever bit of molecular engineering they called the Hydralazine probe.
The Hydralazine probe. Okay, break that down for us.
Think of it like this. Normal hydralazine is like a plain key. If it's a lock, but you can't see which lock it went into, what they did was synthesize hydralazine with a special chemical handle attached to it.
A tag.
A tag, exactly. This handle doesn't interfere with the drug's function at all. But after the drug binds to its target protein, the researchers can use that handle to chemically tag the protein, like attaching a tiny little beacon to it.
So it's like a GPS tracker for the drug to see where it goes and what it interacts with.
That's a perfect analogy. They treated cellosates, basically, the guts of the cells, with this Hydralazine probe. After it bound to its targets, they used the handle to pull out all the proteins that had been tagged, and then they identified them using mass spectrometry.
Which is the gold standard for identifying
This
molecules.
is the highest resolution method we have.
And the result must have been pretty immediate.
It was stunning. Out of thousands of potential proteins, they found essentially one high-occupancy target, the smoking gun, an enzyme called 2-aminoethanethiol dioxygenase.
Just call it ADO.
Yes, please, let's call it ADO. That was the key player. It was the only protein that the probe labeled with high specificity, high occupancy. It was clearly the primary target.
And this is where the big intellectual leap happens, right? This is what connects the blood pressure drug to the brain tumor.
Precisely. Because it turns out that elevated ADO expression and activity had already been independently linked to two very different conditions. The severity of preeclampsia.
The condition the drug treats.
That is in the malignancy grade of glioblastoma tumors.
Wow.
It's not a coincidence. It's the molecular nexus. It suggests ADO is this fundamental, pivotal piece of the disease progression in two completely different systems.
So what is ADO's job in the cell? Why is it so important to the cancer?
Fundamentally, ADO functions as a critical iron-dependent oxygen sensor. This is so important. Remember we said GBM tumors are hypoxic?
Right, low oxygen.
ADO is the protein that senses that low oxygen environment. When oxygen is scarce, ADO ramps up its activity, and it kicks off these signaling pathways that help the cell adapt and survive.
So by targeting this oxygen sensor, the drug is basically throwing a wrench into the tumor's most critical survival mechanism.
You're crippling its ability to thrive when it's being starved of oxygen.
Okay, so they knew the target was ADO. The next step was figuring out how hydralazine actually shuts it down. And you said it's not just a simple block.
No, it's a really sophisticated, what they called a chemical 1-2
A
punch.
1-2 punch, I like that.
So step one is competitive inhibition. ADO, like a lot of enzymes, needs a helper molecule, a cofactor, to work. For ADO, that cofactor is iron, specifically ferrous iron, F2+.
And hydralazine has a structure that lets it just grab onto that iron.
Exactly. It's a process called chelation. The drug molecule forms these really strong bonds with the iron atom, and it physically pushes out the water molecules that are normally sitting there.
So it steals the enzyme's essential tool.
It does,
Yeah.
and in doing so, it occupies the enzyme's active site, which prevents the natural substrate from getting in. That's the competitive part, but that's reversible.
Right, but the real power comes from step 2, where it becomes permanent.
That is the irreversible knockout punch. So the iron center, now bound to hydralazine, kicks off a radical chemical reaction. This causes the hydralazine molecule to fragment, and then one of those fragments covalently attaches to the enzyme itself.
Covalently. So that's a permanent bond?
It's welding the key into the lock. A covalent bond is one of the strongest chemical bonds there is. It's a permanent physical change to the enzyme.
So it converts hydralazine from a temporary inhibitor.
Into an irreversible mechanism-based inactivator. The enzyme is permanently toast, and that's critical for a sustained therapeutic effect.
And this specificity, that's key for avoiding side effects, right? It's not just shutting down everything.
It is, and the data showed it. They found this action was remarkably selective, over 100-fold more selective for ADO compared to other related iron-dependent oxygen sensors.
So it's very good at only hitting its intended target.
Which is exactly what you want in a drug. You want to minimize the chances of disrupting other vital cellular processes.
Okay, we know how hydralazine blocks ADO. Let's really drill down on why blocking it cripples a glioblastoma cell. What is ADO actually doing for the tumor?
So in that hypoxic, low-oxygen core of the tumor, ADO is running on all cylinders. It's constantly activated, and its job is to catalyze the oxidation of certain proteins.
Okay, oxidation. What does that mean in this context?
Think of it as applying a molecular kill tag. This specific type of oxidation is a signal for the cell's garbage disposal system, the ubiquitin proteasome system. A protein gets this tag, and it's sent to be destroyed.
And the specific proteins that ADO is tagging for destruction, are these RGS proteins?
Yes. Regulators of G protein signaling, specifically RGS4 and RGS5. Now, these proteins are the natural brakes in the cell. Their job is to shut down critical signaling networks.
So when ADO destroys the RGS proteins?
The brakes are gone.
Yeah.
The cellular signaling is rewired to favor tumor cell survival, proliferation, invasion. By destroying the cell's own regulators, the tumor gains a huge advantage.
So ADO is basically helping the tumor silence its own internal safety checks.
Precisely.
Hmm.
And we even see it in the metabolic byproducts. ADO's activity produces a compound called hypotaurine, and that compound has been independently identified as a biomarker for GBM malignancy and a poor prognosis. So blocking ADO hits the tumor on multiple fronts.
Okay, now let's circle back. Let's apply this to the 7-year-old blood pressure mystery. If ADO degrades RGS proteins, what happens when hydralazine blocks ADO and lets those RGS proteins build up?
This is the really elegant part of the discovery. When hydralazine comes in and permanently disables ADO, it causes a rapid stabilization of RGS4 and RGS5. They stop being degraded.
And they saw this happen quickly in the lab.
Very quickly. They saw the accumulation in cells in less than an hour, which perfectly matches the rapid drop in blood pressure you see in a patient during a hypertensive crisis.
So by stabilizing RGS, you're putting the brakes back on the signaling pathways that make muscles contract.
Correct. RGS proteins shut down G-alpha-Q proteins. Those G proteins are what tell your vascular smooth muscles to tighten up. With RGS stabilized, that tightening signal gets deactivated much, much faster.
And the functional result is less calcium.
A significantly reduced mobilization of intracellular calcium. And since high calcium is the direct trigger for muscle contraction, reducing that calcium response directly explains the vasodilation.
The mystery is solved. The ghost is finally tangible.
It is, but that brings up a really critical question. If this is so effective at lowering blood pressure, what about using it in glioblastoma patients who might already be quite frail?
I was just thinking that. The dose you need to treat cancer could cause a dangerous drop in blood pressure.
It's the primary safety concern. It's something that has to be addressed very carefully in clinical trials. Now, the researchers do hypothesize that the dose might be manageable.
Why is that?
The GBM cells in the lab actually showed greater sensitivity to hydralazine than non-cancerous cells did. So if that holds up in people, we might be able to target the ADO that's overexpressed in the tumor without hitting the systemic ADO function quite so hard.
But any trial is going to need very careful monitoring.
Incredibly careful. Dose escalation studies with oncologists and cardiologists working side by side. No question.
And this mechanism also explains some of the other weird clinical observations, right? Like the preeclampsia link.
Yes. In preeclampsia, RGS5 levels are naturally lower, so hydralazine, by stabilizing RGS5, helps correct that underlying issue. It even helps explain the rare lupus-like syndrome side effect.
How so?
The research showed hydralazine reduced something called ERK phosphorylation, and that specific alteration has previously been linked to lupus pathology in T cells. The mechanism explains the good, the bad, and the mysterious.
Which brings us to the therapeutic payoff. Hydralazine isn't killing the GBM cells like traditional chemo, it's forcing them into retirement.
Right, this is cellular senescence.
And that's fundamentally different from apoptosis, which is just cell death.
It is. Apoptosis is an execution. Senescence is a state of stable, irreversible growth arrest. We call it cytostatic, meaning it stops division, rather than cytotoxic, which means killing the cell.
For a disease as relentless as GBM, forcing a permanent lockup seems like a really powerful strategy.
It's a huge idea. And the evidence from the lab cultures was very encouraging. They tested it on standard GBM cell lines, U87 and LN229.
And what did they see?
In both, the treatment effectively stopped growth for almost 2 weeks. The IC50, the concentration needed to inhibit 50% of growth, was about 10 micromolar. And importantly, they saw that tumor-type selectivity.
Meaning the GBM cells were more sensitive than other cells?
Yes, more sensitive than non-cancerous cells, and even more sensitive than a very aggressive breast cancer cell line they tested. It suggests that this ADO pathway might be a unique vulnerability in glioblastoma.
And the cells physically changed.
They did. They showed all the classic hallmarks of senescence. They got bigger, they flattened out, and they lost that aggressive clustered behavior.
The physical change is one thing, but you need the molecular proof that they've really entered that permanent retirement state.
And they found it. The biggest molecular signal was a sharp upregulation of a protein called P21.
P21, what does that do?
P21 is a critical break on the cell cycle. Its job is to enforce cell cycle arrest. If P21 levels are high, the cell physically cannot enter the division cycle. The engine is stalled for good.
And they also saw the release of these SASP markers?
The SASP, right. The senescence-associated secretory phenotype. It's the cocktail of molecules that senescent cells release. They don't just sit there quietly. They sort of broadcast their status.
What kind of things do they release?
They found increased expression of inflammatory molecules like IL-6 and IL-8 and other things. These molecular signatures are the definitive proof of that stable, retired state.
And the permanence of this is a huge deal against a cancer known for coming back so quickly.
It's maybe the most compelling therapeutic finding. The experiments showed that a single brief exposure to the drug was enough to trigger the state. When they washed the drug away, the cells stayed arrested.
So it's a one-way trip.
A one-way trip to permanent arrest. For a disease this invasive and resistant, that is a profoundly effective maneuver.
Okay. We have a confirmed mechanism that induces permanent tumor arrest. That's revolutionary. But now we have to talk about the roadblock, the biggest challenge in all of neuro-oncology.
The blood-brain barrier, the BBB.
The biological fortress.
It really is. Its job is to protect the brain's delicate environment from everything, pathogens, toxins, chemicals. It's formed by these specialized brain endothelial cells that are stitched together by something called tight junctions.
And these junctions are much tighter than in blood vessels anywhere else in the body.
Incredibly tight. They eliminate all the little gaps. But it's not just a physical wall, it's also an active gatekeeper.
It has a surveillance system.
It does. The BBB is filled with these active efflux transporters. They're molecular pumps, P-glycoprotein, MRPs, BCRP. These pumps recognize drug molecules, grab them, and just pump them right back out into the bloodstream.
And the net result is that almost no drugs get through.
The number is staggering. It's over 98% of all small molecule drugs are excluded from the brain. The complexity of getting past that barrier is why progress against GBM has been so painfully slow.
So given that hydralazine is not a drug that easily crosses the BBB, how did the researchers prove that this mechanism even works inside the brain?
They had to do a conceptual bypass. For their mouse models, they used what's called intracerebroventricular injection.
Meaning they injected it directly into the brain.
Directly into the brain's ventricular system, yeah. It completely bypasses the blood-brain barrier.
Okay, so that proves the target, ADO, is there in the brain and the drug works on it. But it doesn't solve the delivery problem for people.
Not at all. The analysis of the brain tissue from those mice confirmed ADO was the only high-enrichment target, so the interaction works perfectly once you're past the barrier. But it also proves that the current generic form of hydralazine is just not going to work for systemic treatment in humans.
Delivery is the limiting step.
The immediate critical limiting step. The next stage has to be all about medicinal chemistry, about creating a brain-penetrant version of this drug.
And the good news is they have a blueprint, right, from the crystal structure.
An invaluable blueprint. They have the crystal structure of hydralazine bound to ADO. That lets them see exactly which parts of the drug are essential for binding and which parts they can. Well, they can modify.
So they can tweak the non-essential parts to help it get across the barrier.
That's the goal, to improve its ability to pass the BBB.
And what are the specific properties they need to optimize for that?
It's mostly about two things. Lipophilicity, which is how well it dissolves in fat and molecular weight. Drugs need to be small, generally under 500 Daltons, and they need to be lipophilic enough to cross the fatty membranes of those endothelial cells.
And hydralazine, as it is too polar. It's not greasy enough.
That's a good way to put it. So the medicinal chemist's job is this tightrope walk. Make it greasier without messing up the part that actually targets ADO.
Are there other strategies besides just changing the molecule?
Oh, yeah. There's what's known as Trojan horse delivery. This is where you leverage the brain's own transport systems.
How does that work?
It's a process called conjugation. You link the drug molecule to another molecule, a ligand that the brain actually wants. You're basically attaching a shipping label that says urgent delivery to the brain.
So you could link it to something like transferrin or insulin.
Exactly. Things that target receptors that are already highly expressed on the PB. The receptors mistake the whole package for a nutrient and actively pull it across. And then there's nanodelivery.
Encapsulating the drug in tiny particles.
Specialized nanoparticle formulations, yeah. These can sometimes slip through the barrier, or they can be designed to release the drug only once they're inside the brain.
Now, there is a twist here. The tumor itself, glioblastomas, are known to kind of disrupt the blood-brain barrier. Does that help?
It creates a fascinating paradox. The blood vessels inside a glioblastoma are abnormal and leaky, so that might allow more of the drug to passively seep into the tumor compared to healthy brain tissue.
So that leakiness could provide some natural tumor targeting.
It could. But that's the two steps forward part. The two steps back part is that the tumor counteracts this.
How?
The blood-tumor barrier, despite being leaky, paradoxically has an enhanced expression and activity of those very same efflux pumps.
So it's leaky, but it's also actively pumping out anything that gets in.
Exactly. The tumor is simultaneously inviting things in and then hyper-efficiently kicking them back out. So the challenge is designing a drug that can take advantage of the leakiness while also evading those super active pumps.
Despite these hurdles, this discovery really highlights the huge strategic value of drug repurposing, especially in oncology. For a disease like GBM, the timeline is everything.
The comparison is just stark. Developing a totally new drug from scratch takes, on average, 10 to 15 years and costs over $2.6 billion.
And most of them fail.
More than 90% of them fail once they get into human trials. It's a massive, slow, high-risk gamble.
Whereas repurposing, it de-risks the whole process from the very beginning.
It completely collapses the timeline and the cost. You get to skip the entire preclinical safety assessment, the toxicology, the basic metabolism studies, because all that work has already been done.
Which can shave years off the development time.
It can reduce the timeline by 3 to 12 years and cut costs by more than half. For patients with a 10 to 13-month median survival, skipping a decade of safety trials isn't just an economic benefit, it's a moral imperative.
And hydralazine is just a perfect poster child for this strategy.
It really is. 70 years of clinical experience. We know its safety profile inside and out, even in vulnerable groups like pregnant women. It's stable, it's cheap to make, it's widely available, and it's a generic.
That existing safety data also must give it a huge leg up in the regulatory process.
It does. Agencies like the FDA have these expedited development programs for serious conditions with unmet needs, which GBM obviously is.
So it could be a candidate for fast track or breakthrough therapy designation.
Yeah, it could. And if they can show it's working by monitoring a surrogate endpoint, like that P21 upregulation in the tumor, It might even qualify for accelerated approval.
Okay, but this is where we hit the big economic dilemma, the tragedy of the commons.
This is the critical financial hurdle for generic drugs. Since hydralazine has been off patent for decades, there's no patent protection left. Pharmaceutical companies, which are for-profit, have basically zero financial incentive to spend the hundreds of millions required for massive clinical trials.
Because if they spend all that money to prove it works for GBM, every other generic manufacturer can immediately start selling it for that use without spending a dime on the research.
They get to free ride on the approval. That's the dilemma. The drug is scientifically promising, but it's too cheap, and the research is too expensive. It's a classic market failure.
So what are the solutions? How do you incentivize that investment?
Well, there are some policy tools. One is a method of use patent. This gives you exclusivity only for the new indication, just for treating glioblastoma without trying to repatent the molecule itself.
And there's also regulatory exclusivity.
Right. The FDA can grant a 3-year exclusivity period for drugs approved for new uses. But the question is 3 years enough to recoup $1 billion investment?
Probably not.
Probably not. Which is why this highlights the growing need for public-private partnerships. Government agencies, cancer-focused nonprofits, academic centers, they have to step in to either fund the trials or at least mitigate the financial risk for industry partners.
Okay, so the scientific evidence is compelling, but it's all in the lab right now. What are the next critical steps before this could ever be tested in people?
We need deep, sophisticated preclinical validation that really accounts for the complexity of human GBM.
So moving beyond the simple mouse models, what are the gold standards now?
First, PDX models. That's patient-derived xenograft models. You take actual human tumor tissue from a patient's surgery and implant it in a mouse. This lets you test the drug against the real genetic chaos you find in a human tumor.
Okay, what else?
Second, GMMs, genetically engineered mouse models. These are mice that are engineered to develop these tumors on their own. This lets you study the entire tumor life cycle and see if ADO inhibition can slow the disease from the start.
And finally, a model that simulates the brain environment.
Exactly. Orthotopic models. That's where you implant the tumor cells directly into the mouse brain. This is crucial for recapitulating that low-oxygen microenvironment and for really assessing how the drug interacts with the blood-brain barrier. No human trial happens without this battery of tests.
And once we get to human trials, doctors need a way to know if the drug is actually working inside the patient's brain, right? Before a tumor might shrink on an MRI.
Biomarkers are absolutely critical. We need a reliable way to confirm that ADO is being inhibited.
And you can't just keep taking biopsies of the brain. You need a surrogate.
Exactly. One idea is to use PBMCs, peripheral blood mononuclear cells, from a simple blood draw. If you can show the drug is stabilizing RGS4 and RGS5 proteins in those blood cells, that's a good non-invasive sign that it's active in the body.
And for monitoring the tumor itself.
If you do have tumor tissue, you can look for those senescence markers like P21, but we can also use advanced imaging. There's a type of PET scan, 18FF PET, that tracks metabolism in GBM. If the drug is forcing cells into senescence, you'd expect to see metabolic changes that this kind of scan could pick up.
And the trial design itself will have to be unique because of the cardiovascular effects.
Absolutely. Phase I trials will be all about safety, finding the right dose in patients with recurrent GBM, but these trials must be co-monitored by both oncologists and cardiologists, working together to ensure patient safety.
Now, given how tough GBM is, hydralazine will probably be used in combination with other therapies.
Almost certainly not as a standalone. The most exciting idea is that 1-2 punch, senescence strategy.
So hydralazine is the first punch, forcing the cells into arrest.
And then you come in with the second punch, a senolytic agent. That's a drug designed to selectively kill only those cells that are in that senescent state. So you arrest the tumor, and then you clear out the arrested cells.
And what about combining it with existing treatments, like chemo or immunotherapy?
It could be combined with temozolomide, but the immunotherapy angle is really exciting. Remember the SASP, that cocktail of inflammatory molecules senescent cells secrete?
The inflammatory beacon.
Exactly. That beacon can actually attract the immune system, making the tumor more visible to T cells. So you could combine hydralazine with an immunotherapy, like a checkpoint inhibitor, and potentially turn a cold tumor into a hot one that the immune system can attack.
And finally, the implications here go way beyond just glioblastoma.
The initial data suggests they do. High ADO expression also correlates with poor prognosis in several other very tough cancers: liver, cervical, pancreatic cancers. This discovery validates ADO as a brand new, widespread target in oncology.
This has been a really incredible deep dive, but as we close, it's so important that we provide a really clear, sober message about where this research stands right now.
Absolutely. While this offers genuine cautious hope, we have to issue a critical warning. Hydralazine for glioblastoma is entirely experimental and investigational.
There have been no human clinical trials for this use. All the evidence we've talked about is from the lab and from animal models.
So for anyone listening who is dealing with this diagnosis, you should absolutely not attempt to self-medicate or change your prescribed treatments based on this. You must consult your neuro-oncology team.
We can't overstate that.
The safety concerns are severe. Using this drug outside of a controlled trial could cause dangerous, even life-threatening cardiovascular side effects. It could interfere with your other therapies. This is a potent drug that needs careful supervision even for its intended use.
So for anyone interested, the only safe pathway is to monitor for enrollment in properly designed clinical trials, if and when they become available. A resource like clinicaltrials.gov is the right place to look.
At the end of the day, this discovery validates ADO as a major druggable target. It's just a brilliant example of how fundamental research, even on an old sheep drug, can unlock these profound opportunities.
The story of hydralazine reminds us that sometimes the most revolutionary advances aren't found in brand new molecules, but in deeply understanding the therapies we already have, which just makes you wonder, what other secrets are waiting to be discovered in our medicine cabinets?
The enduring power of curiosity-driven science. The mystery of the past becomes the treatment of the future.
That was our deep dive into the hydralazine breakthrough here at the Urban Herald. Thank you for joining us.